And thanks for the introduction. So, as most of us here know, genes are regulated by possibly dysregulatory elements called enhancers. And our goal within this work is to find enhancers, link them to target genes, and use them to predict gene expression. And there were various different methods developed for this specific task. And here are some examples that we can divide into two groups. And one of the first group is contains the sequence-based approaches, where we have the DNA sequence as input, and as output we have the predicted epigenetic signal tracks or gene expression. And then on the other side we have the gene-specific approaches, where we have the epigenetic signal tracks as input, and as output we have the predicted gene expression or important regions for a gene. So within this project, we chose to develop a gene-specific modeling approach, and also we want to use single-cell data. And here you can see some approaches that are gene-specific and that use specifically single-cell RNA-seq and single-cell attack-seq data. So these approaches can be subdivided into univariate approaches, where we have the individual modeling of peaks and genes. And on the other hand, we have the multivariate approaches, where we regress over all regions within the window around the target gene. And we were also interested in using a multivariate approach, because we're not only interested in modeling the dependency between regions and a gene, but also between regions. And the best state-of-the-art method for this currently is ScarLink. But ScarLink has the limitation that it is only able to model activating regulatory effects. So since we're dealing with single-cell data, and it was already mentioned in the talk before, we deal with inherently highly sparse data. So a typical single-cell RNA-seq matrix has around 90% zeros, and a single-cell attack-seq matrix even 95%. So it's really difficult to extract biologically meaningful information from this. And a common strategy is here to aggregate the cells into metacells, so basically cluster cells according to their similarity in their gene activity profiles. There are also various approaches for this. And we are also interested in this work to see actually the impact of this signal aggregation on gene expression prediction. So our exact learning setup here is we have a window around a gene. Here we use a window size of 1 MB, and within this window, we bin the genome into equally sized bins that hold the quantified epigenetic signal for each sample. And this we use as features to predict gene expression. And in theory, we could use any kind of machine learning model here, but we're training one model per gene. So when we have 20,000 genes, we're training in the end 20,000 models. So we need to choose a model that is actually computationally feasible to do this. So we developed a fully automated Nextflow pipeline called Metapher. And as input, we have multi-ohm RNA and attack data. So RNA and attack data measured in the same cell. And in the next step, we have an optional metacell creation step to address the sparsity problem of the single-cell data. But when we use this metacell aggregation, we can also use unpaired data as input. And I will show you the exact method that we're using on the next slide. Then we can generate our feature matrix for each gene. So again, here we have within each bin the quantified attack signal for each sample cell or metacell. And then we train a random forest regression approach to predict the gene expression for each gene. And we chose the random forest regression because it's a very simple and fast model. And it also is able to capture nonlinearities. So the metacell creation method that we're using was developed by our former lab member, Fatime Vejati. And it is an available R package that is called Metaceller. And there are two major steps. So the first one is clustering the RNA cells into metacells using a K-medioid approach. And the second step is assigning the attack cells to the closest RNA metacell. And here we cluster the RNA cells by cell type or any other metadata that you want to give to the clustering algorithm to preserve the underlying data structure. And an important parameter here is the K, so the number of cells per metacell, because this in the end also impacts the number of training samples that you can use for model learning. Yes, so what I wanted to say as well is that for the unpaired data that you can use here in this approach, it needs to be integrated. So the integration is not part of a pipeline. Okay, so now to our first results, the impact of the metacell aggregation on model learning. And here you can see, so we applied our pipeline to the 10x PBMC test set, multi-ohm data set that comprises around 10,000 human blood cells. And as a test metric, we calculated the Spearman correlation between the predicted and the actual expression. And here you can see the results for a different number of cells per metacell. And what we can see is that initially, when we increase from 100 cells per metacell, from 50 to 100 cells per metacell, the test correlation increases, so the performance gets better. But then it reaches a plateau because, I mean, this can then be explained with the number of training samples, because naturally, the number of training samples decreases when you merge more cells together. And you have this trade-off that you want to have the most high-quality samples, but also you want to have enough samples for model training. So then we also wanted to compare the performance of our metacell setup to the single-cell setup. And here, for the metacell setup, we used the 100 cells per metacell then, and we have our trained single-cell models and our trained metacell models, and we predict the expression on a 20% test set. And so for the single-cell setup, there's an additional step where we sum the predictions for all cells that belong to a metacell, and then we calculate again the test correlation. And here you can see the results. So on the x-axis, we have the metacell setup, on the y-axis, single-cell setup. And what we can see is that for the majority of genes, we actually outperform the single-cell setup, but there's still a substantial amount of genes where the single-cell setup is better. But overall, the metacell setup increases the performance. So then we also wanted to check if there are any differences for the single-cell and metacell setup in their ability to find biologically meaningful interactions. So what we first wanted to do, we wanted to find the important regions for a gene. And here we use the explainable AI method, SHAP, that calculates the contribution of each feature to a model's prediction. And we use then the SHAP value to rank the individual region gene pairs across all genes. And we also used a distance decay weighting. Then we obtained whole blood fine-mapped EQTLs from GTEx, and we asked ourselves if we could find these EQTLs, if these are enriched in our top-ranked interactions. And so here you can see for the single-cell and the different metacell setups, the enrichment for the top 4 most abundant cell types in our data set. And we can see that for the metacell setups, the median enrichment is higher than for the single cell. Then we also benchmarked our method against the state-of-the-art method SCALing. And so SCALing actually has the exact same feature setup. So we also have a window around the gene, and we have the binned quantified attack signal for each sample, and we learn 1 model per gene. The difference is the machine learning method that was used here, and here we have a regularized Poisson regression approach. And so for running both methods, we had for SCALing an initial gene set of 2,000 genes. These were the top variable genes. And we could, in the end, obtain a model for around 600 genes. And for Metapha, we actually did not apply any initial filtering on the gene expression. And we could obtain a model for around 19,000 genes. And here we looked at the mean squared error between the predicted and actual expression on the 500 common genes where both methods could obtain a model. And we can see that Metapha outperforms Scarlink on the majority of genes. And again, when we look at the Spearman correlation, so here you can see actually for both methods, the number of models where both methods could generate a model with correlation above 0.3. So for Metapha, it was around 600 genes, and for Scarlink, around 320. We ran both methods on CPU with 25 cores, and here you can see the runtime per gene in minutes. So Metapha processes a gene in about 6 seconds, and Scarlink took around 4.5 minutes. So in the end, for processing the initial gene sets, Metapha took around 1 day and Scarlink around 6 days. But I have to say here that Scarlink was actually optimized to be run on GPU. Finally, we also wanted to see if gene features have an impact on our ability to predict gene expression. So basically what we did here, we classified our genes into 3 classes according to the test correlation in failed, medium, and high. And the first gene, and then we stratified by gene feature. And the first feature we looked at is the expression sparsity, so the fraction of zeros in the gene expression. And again, we can see here really nicely that it is negatively correlated with our model performance, which is also cohesive with our previous findings that metacell aggregation indeed increases our performance. And then we also found that the number of transcription start sites, here more specifically GenCode annotated transcription start sites, is positively correlated. And so finally, to summarize our findings, we developed a fully automated Nextflow pipeline called Metapha to predict gene expression using single-cell epigenetics and transcriptomics data. And it was successfully applied to human single-cell data set. And we showed that summarizing the cells into metacells improves model prediction performance. And we were able to outperform the state-of-the-art method in both accuracy and runtime. And finally, I would like to say thank you to the Institute for Computational Genomics Medicine, especially Marcel Schulz for his great supervision, and Dennis Hecker and Fatima Bijati, who are also involved in this project. And thank you to the funding, and thank you for your attention, and I'm happy to take your questions.
Can I have a small announcement? Because of the overflow, we are so happy that many of you wanted to come. So rooms 6 and 7 are set up and streaming the presentation that is happening right here, right now. Rooms 6 and 7. Okay.
Thanks for your talk. Very interesting. Hi. Yeah. I was wondering, so you mentioned that some genes performed better with your single cell setup and most genes performed better with the metacell setup. Are there any characteristics of the genes that perform better in either method? Like, are they more conserved? Are they involved in certain pathways? Or was there anything you could see there?
This is a really interesting question. And actually, it was quite last minute that we started to look into the gene features. And this is definitely something we have to explore in the future.
Okay, thanks.
Given a completely unrelated NIV ataxic data, how well can it recapitulate RNA expression values?
Well, this was now trained on the blood cell data set. And so we used a random 20% test split. But yeah, of course, also all these cells are highly correlated, even, yeah. But we also plan to generate a larger data set where we have many more cell types and then use this for model training. And then we can also work on the generalizability of this.
Hello, thank you for your talk. I have two questions. First one is maybe I misunderstood. Did you apply SHAP to your model inputs, or?
SHAP was used to rank our features basically in the end for each gene.
As in your like meta cell attack input feature?
So here we use SHAP actually for the single cell setup. But yes, I mean, it doesn't matter if it's meta cell or single cell. So basically in the end, when we have our trained models, so it's after the model training that we want to get the feature.
My question is, if your trained model is random forest has like a very simple, a very reliable method.
The feature importance.
So why didn't you use that and resorted to SHAP?
I mean, we were thinking of also, I mean, the random forest is not like the ultimate model. And of course, we want to try other models in the future. And that's why we used also SHAP to.
Okay, yeah. Thank you. And my second question is, did you check the impact of read depth on your results? Because some neighborhoods where you combine cells might have higher RNA-seq read depths and others slower. So how did it affect your predictions? Thank you.
I mean, we're normalizing the data, so it's not on raw counts. I'm not really sure if I'm getting the question. So, I mean, we're basically using the meta cells to aggregate the data to get more read depth overall. Yeah, okay, we talk.
Thanks, Laura, for a nice presentation. So we've been also thinking quite a bit about meta cells, sort of starting some stuff early on. Not sure if this is sort of a crux because we want to just reduce, sample numbers, sort of up read depth. I mean, the ultimate meta cell is just pseudo barking, right? Or even just fully barking, which we know sort of has all kind of additional statistic errors. So do you think maybe we should sort of be more fine-tuned in maybe some of the downstream methods, the sort of that deal automatically with like a local barking, but sort of have an average window? Because, obviously you sort of at the fringes, your cells at your pseudo bark or meta cell or whatever, right? They're aggregated away. So do you think in the long run, you still have this sweet spot of, let's say, aggregate 100 cells or whatever? I think you found something like that. Maybe also dynamic version, or maybe on the long run, we just go really full single cell if we sort of adopt the downstream statistics.
Yeah, I mean, right now, so there were like multiple aspects. So right now also, I mean, this 100 cells per metasite worked for this data set, and it definitely has to be tuned for each data set because also it's cell type specific, and we're also doing the clustering cell type specific if the data is available. And you can also give other metadata. And I mean, depending on the dimension of the metadata that you give, you also have to tune this parameter. What is nice is that you, for these fast models, you can just try a couple of parameters out. And yeah, but overall, of course, it would be nice to have it on single cell level to really capture the individual heterogeneity. Yeah.
Thanks, Laura.